Mike DiLegge. Oh my goodness, it's so good to have you on MyFlora Fridays. We have been connected for a while. Actually, I think my first interaction with you was in person at the Emerald Conference in San Diego a couple of
Yeah,
years ago. You
2023.
were friends with Tess Edom, and Tess and I had an Airbnb, and you guys had an Airbnb across like this little street from us, and you gave a proper like stage talk. And I think I had a poster and was really nervous because it was my first poster ever, even though like I went through my whole master's and PhD without ever presenting a poster. But yeah, that was our first interaction. And I remember seeing you do your talk about microbes and what you were doing at the time. And I don't know that much about microbes. And I was just like, wow, this is so cool. So I really am so glad that you're on with me because I have lots of thirst for knowledge in your area. And I think you're awesome person. So thanks so much for joining me. But you know what? I don't know you very well. You know you more than I know you. So go ahead and tell the people who you are, what you do, and what brought you to cannabis.
Yeah, right on. Thank you, Anna. I appreciate you having me on. And yeah, so my name is Mike DiLegge. I'm the director of microbiology at Impello Biosciences, and I live in Colorado. So being in Colorado, it was pretty fun and lucky to get into the cannabis industry when I moved out here. I moved to CSU and started studying plants in 2015 and got my degrees in horticulture, first food crop production, and then plant microbe interactions. So we'll talk about microbes here in a little bit. But yeah, growing up on the East Coast and relocating to Colorado, there was obviously an interest for being able to, smoke cannabis and use it as medicine without any penalties from
Hmm.
having the legalization in 2013 here. And then, yeah, when I finished my master's degree at CSU in 2020, I got involved with Impello Biosciences, and we are a company that sells, plant probiotics is what I like to say, or bacterial inoculants for the root zone or even the phyllosphere, the plant leaves and the above ground parts. Being in Colorado, our first clientele for products like that were the cannabis industries because the growers in cannabis just, want to love their plants so much and try to give every good input they read about to the root zone. And definitely things like beneficial microbes can help outcompete things like pathogenic microbes and disease causing organisms as much as we all try to want to, pretend to maintain a sterile cultivation system. There's always going to be microorganisms in those spaces unless you're in like a lab or a super sterile setting. So yeah, cannabis was one of my first lab rats right out of school. I got to experiment with it using different types of bacterial inoculants to see how we could, increase things like THC or CBD, or just kind of help support overall plant growth and plant health.
Okay, awesome. All right, so that's like scratching the surface of where you're at, right?
Right on.
So so I know that a lot of people in cannabis, first of all, we've got the indoor versus outdoor. Obviously, outdoor, you've got natural soil, natural sunlight, all natural airflow.
Mm.
Not to say that outdoor is better, but and then we've got indoor where I feel like most facilities grow in inert conditions, kind of, cocoa or just deep water culture. And so they're growing in media that's sort of devoid of the natural organisms that you would find in living soil. So is that kind of like a big area in cannabis? And is that where you like what do you do? What have you seen? How does this work?
Absolutely, yeah, yeah. Thank you. I mean, mostly, these substrate mixtures, whether it's rock wool or stone wool or peat moss or some combination of all that kind of stuff, it does come with its own natural microbiome. It's just what we like to call is like low microbial biomass systems. So low populations of bacteria and fungi, but like we talked, it's kind of impossible to maintain sterile plant roots because your plants want microbes on their roots. And yeah, trying to populate those substrates with things that we see pop up in natural soils, in living soils, different bacteria associations, bringing plants inside and kind of growing them in a controlled system like that, it does give the grower, the cultivator, a lot more precision on what to do. So you can get different inoculants or brew your own, like compost teas and things like that, and then just try to populate that root zone or that rhizosphere, as well as all these low microbial biomass substrates because they're clean and they're easy to work with. They're not going to run a bunch of particulate matter and clog a filter or a hydro or a recirculation system. But yeah, the part of the reason, like Dr. James White, some of his research shows plants pretty much formed roots because of their relationships with microbes. They really like to cultivate microbes in their root zone. When you allow your plants to find whatever they can in the environment. Sometimes those are disease-causing organisms, sometimes those are beneficials, and sometimes they're just pretty inert. What we've seen is when you can kind of prime the root zone with beneficial bacteria early on and as soon as possible, it's not only a healthy approach to kind of expand the rhizosphere and the root architecture. More strength below ground allows more weight above ground to be produced. Same thing with kind of just an ounce of prevention is worth a pound of cure, that old saying.
Mm-hmm.
It's if you populate that rhizosphere with a beneficial microbiome before things like Fusarium or other disease-causing organisms try to take a seat at the table. One analogy I like to use a lot is the plant roots are an empty dining table, and they make these plants make these sugars called root exudates or other organic acids and compounds that they deposit into the soils or into their hydroponic systems, mostly to recruit different microbes is what the big theory is. It's kind of like a crying out for help. There's a stress, I'm going to exude this compound. A lot of times during younger growth and development, we see plants exude a lot of sugar and carbon because microbes' favorite things to eat are cakes and cookies, which is basically just sugar and carbon and things in there. And then as plants age and start to really pump a lot of energy into the reproductive biomass, a lot of research shows they change their root exudate compounds into organic acids and things like that are a little bit less generalist food sources for all these different kinds of microbes. So bringing it back to the dinner table, if you have a bunch of empty chairs around a table and your plant roots are basically, carrying food out, serving food onto the plate, you want a lot of different kind of compounds to keep a lot of different seats at the table. And the sooner all those seats are full, the sooner the rhizosphere is, populated by different types of beneficial microbes, the less chance there's going to be an extra seat for something like Fusarium or Pythium to move in and start to kind of feed off those root zones too. So the sooner you can populate that table with things that are either going to be beneficial or inert or helpful for your system or your plant or both, there's less room for things like pathogens to come in and cause infection.
So like a really popular restaurant that serves awesome food is like really hard to get into brunch on a Sunday morning. There's people
Yeah.
like waiting outside, and if there's no seats, there's no seats.
Yeah, think about hungry fusarium kind of pounding its foot at the door, waiting to be let down. And we're just too busy today. We got too many beneficials sitting at the table. It's kind
Go
of
away. You're gonna have to come back some other time.
and another cool point when we move back, outdoors out of the greenhouse into these living systems or living soil systems or protected agriculture greenhouse where there's just soil on the bottom that's outdoors. You got people I'm sure hear about permaculture practices, things like companion planting, whether that's co-planting cannabis with wine cap mushrooms or co-planting cannabis with clover or something like that. Different
Hmm.
plants will provide different food for that table, right? So if you kind of alternate, considering the space for your host to remain productive, but if you alternate or even kind of crop rotate, you can put different foods at that table to allow, a diverse set of competition on the microbiome, on the rhizosphere there. And, keeping, but mostly, it's a great thing to keep pathogens away, but then you'll start to find out your guests at the root zone start to benefit the host too. They can unlock nutrients like phosphorus or, iron uptake. These essential plant nutrients are only the microbes don't want to stop eating. They want to sit at the table forever. So as long as the host gets tips or benefits from the nutrient conversion in the soil or in the root zone, then it's kind of like a symbiotic relationship.
Like, but as long as they were like kept ordering foods and drinks
Yeah.
and like, they're gonna tip the servers well, like nobody cared. But there's also restaurants where you want like that table turnover you want.
Turnover.
So
Yeah.
yeah. So I don't know if that's like quality over quantity situation that kind of
Yeah.
could be
Yeah.
translatable to your analogy.
Another kind of analogy people like to use that's really similar to the one I like is an auditorium at like a sports arena or something. So rather than kind of camping and keeping all these people feeding, which might not be great for the server or for the host, or it could be, articulated that way. Think about like a really entertaining sports event or a theater performance or something that's kind of captivating those audiences. So they're camping and staying around too.
So you mentioned Dr. James White. I know James. He is up here at Rutgers University,
Hmm.
and I have been to up there and visited him. I've talked to him a few times about various things. We were going to do some research with one of his grad students, April, that I don't know, I feel like she may have had a lot going on, and we never actually made those experiments come to fruition, but we had an idea to like, and I know, like, again, microbes are not my strong suit, but he had some really interesting research, and she had some really interesting research about using microbes to increase various compounds that we're interested in, such as THC or terpenes. And I'm not sure how that works, but we were going to inoculate, like, have, different, the same strain, but inoculate them with different combinations of microbes
Mhm.
to see how the plant reacts to those interactions with those various combinations. So I know this is not, maybe it is your research, but I bet you know a whole lot more about that than I do. But can you kind of explain how that works?
Yeah, yeah, I can, do my best. I'm nowhere near Dr. James White's level of microbial expertise. Yeah, yeah. I think my first actual exposure to him was on your other podcast, the one from a few years back,
The tank
the Dank
hour.
Hour with everybody. Yeah, yeah. But yeah, I mean, so some ways that microbes can change their plants or their host metabolites, right? The secondary metabolite synthesis would be to kind of function as adjacent to like a plant growth regulator or PGR. They can activate a pathway ISR or SAR, so induced systemic resistance or systemic acquired resistance. And those are two different pathways. They run along the plant defense pathway. Things like, I think it's the salicylic and the jasmonic acid pathways are the primary reactive pathways when one of those things gets triggered, the ISR or SAR.
Uh-huh.
So when you apply these microbes in different combinations, especially when there's more than one in a controlled system like a lab research experiment, it allows for basically like a two or three species conversation that's happening on the root zone, and different peptides or microbial produced proteins can, without damaging the host, kind of trigger an upregulation of defense compounds, right? We love plant defense compounds, THC, CBD, CBN, THCP, all the different cannabinoids. They are some version of defense compounds. I think I've even read some papers where if a cannabis plant gets infected with aphids, while nobody will want that near the flowering season, it will upregulate its THC synthesis and stuff like that as a defense compound to become bitter or less palatable to chewing and biting and sucking insects and whatnot. So in a similar way, but without damaging the plants, microbes can activate these pathways, triggering them at the root zone, and then kind of having a reaction in the host. One of my favorite things in microbiome research is the overlap with all the methods of the way people look at these communities. So whether you're like a lichen on a high alpine mountain or a cow gut or a people gut or plant roots, it's really similar data analysis. And these ecology, these like microecology community interactions are super neat. But yeah, one of my favorite another analogies, I'll do that a lot and maybe personify way too much for a scientist.
No, I
But
like it. I like it.
I mean, me and I'm sure a lot of other people as well like to call the plant roots basically like the plant's gut, right? We can take, they say like 70 to 80, maybe it's even 90% of our serotonin and dopamine as humans is first starting to, upstream at the plant gut brain or at the gut brain barrier, not the plant. We're talking about people now. And I think in a really similar way, these induced systemic resistance pathways or the systemic acquired resistance pathways start at the plant's gut in the roots. They're responding to a signal and then kind of changing their internal chemistries like we would with our brains and happy hormones.
I always feel like when when we're talking about plant defenses and using them to make the plant do something that we know that it's going to do because it's in response to something that it's perceiving as a threat,
Mhm.
I kind of feel like it's torture. We can't talk to plants, right? And I always talk about, the smell of fresh cut grass is not like we like it, but it's really
Mhm.
the smell of screaming. It thinks
Yeah.
it's being decimated by locusts or something, and it is like letting off all these chemicals to let the other plants around it know, like, oh my God, like something terrible is happening. And I think of it like torture.
Yeah, I mean, think about a lawnmower. That's definitely a torturous environment, and the neighboring lawns are responding to those volatile signals and the grass screaming, right? And probably making themselves taste a little bit worse with something like a locust. I mean, I see what your point. That's a good point. You know, Another way to look at this, though, at least kind of maybe the happy middle ground between like mechanical damage, like a lawnmower, and microbial pathway upregulation. All plant immune systems have things called MAMPs and PAMPs and DAMPs. It's like disease associated pathways, microbial
Yeah.
associated pathways, mechanical damage, pathogen associated. I kind of like to think of like the microbiome or the rhizosphere, as long as it's not a disease causing one or something that's going to stress the host out too much, could be something like a maybe more of like a verbal signal. It's not causing disease. It's not a caterpillar chewing on a neighboring plant leaf or anything. It is just kind of starting to colonize the rhizosphere and upregulate these different pathways. But yeah, I mean, if it's defending itself, it might be scared. It's a good
Or
point.
like maybe like athletic training. Because, I hate running, but I know that it's good for me.
Same.
It'll make me stronger. I'm probably not gonna run, but like maybe I could do some strength training that's not as difficult on my lungs.
I like athletic training. That could be a good way to put it. I always like to say I identify as a smoker, not a runner. Uh, but yeah, yeah, it is kind of like these small signals, like kind of like maybe lifting weights, right? You're ripping your muscle tissue, these little tiny fractions, and then as they heal, they get bigger and more trained, right? A priming of a response before the stress is induced. Yeah, like so kind of filling up the dinner table before Fusarium comes to sit down is also kind of like training
Mhm.
training how to maintain a balanced rhizosphere, right?
Yeah. But, when you think about a plant, like when you think like a plant, they can't get up and move. They have to be able
Mhm.
to do something at the time that shit's happening. They have to be able to do it quickly in response to whatever's going on in the environment, whether it be good or bad, right? They don't want to
Right.
spend too much energy doing something if the conditions are great. Like, and they can kind of like chill for a little bit. They might want to like take a nap and not do so many things. But if shit's going awry, it's drought or insects or whatever, like they've got to be able to react to that super quick. And there's an experiment that is really interesting to me, that I think it's the mimosa plant that when you touch it it closes its leaves.
Yeah.
They used that plant, and they would take it up a ladder and then drop it, and as soon as as soon as it hit the ground, it would like close up. And they kept
Right.
doing this, and the plant eventually realized, like, oh, this is not harmful, like this is just, I know what's going to happen, and it's nothing. And it responded to being dropped at first, but then it learned that that isn't something that is a threat, and so it stopped spending the energy on closing up all its leaves. And I always think that's so interesting. There's
Yeah, I think
research.
that is really really neat. They get a memory and everything like that. I read somewhere, and I didn't find a sign. I mean, I didn't fact check this or dive into it, but it made sense. It was like when plants are having these like small stressors, everything from the field to your house plants. There's like animals, I think cats specifically can pick up on some of the sounds or frequencies they put out when they're under drought. And that's why cats like to knock over house plants, because when they need a little bit of water before they're wilting, they're already starting to scream. So the cat's just like, "Hey, shut up, get off my counter."
That
um,
is super interesting because my cat, Jean, we have banana plants that, we're in New Jersey, so it's freaking cold now. Bananas don't do well in cold weather, but they
mhm.
look great on our patio in the spring through, the beginning of fall. But we have to bring them inside in order to keep them and keep them alive. And
Right.
she will sit there and she'll stare, and they're big. They're like 6 feet tall. She'll sit there and she'll stare like at the soil for like an hour. And we're like, what are you looking at? And we always thought it was microbes, but maybe she's listening to the plant, because I don't water them that much when they're inside because they're
Yeah.
kind of, but maybe that's my cat listening to the plant. Oh my God, now I feel so bad.
Well, yeah. I mean, if they
And
want to
she's
be
so
dormant
funny.
too, maybe they're making like a
If I go get the
yeah.
jug of water, she'll like follow me and like watch me water them all. She loves it.
Interesting. Maybe she's like, "Hey, we're gonna quiet these plants down together, Mom." That's pretty interesting. Like,
Wow, that's
finally.
cool.
Yeah, yeah, yeah. Like I said, I imagine there's got to be some studies associated with it. This was like a headline, but it was really interesting, and it was satisfying enough for me to be like, "Oh, that's why cats knock over houseplants to be like, hey, be quiet. I'm trying to sleep."
Well, now I'm going to go down that rabbit hole because I know that Brett's super interested in why Jean does this. It's funny. It's funny. And she'll sleep next to the plants too, like in the pot, because they're in big, big
Okay,
10
yeah,
gallon pots or more,
that's awesome.
maybe
I bet they're
15.
beautiful.
They only will grow about have like 5 leaves on them at a time.
Hmm.
They'll let one go and then they'll sprout another one. So they're constantly growing. They do better out, obviously they do better outside, and they grow about a new leaf every week.
Yeah,
But yeah, they've only ever had about 5 or 6 leaves max.
right on.
But in the last, let's see, how old are they? 3 years, I think. I brought two home that were about this big.
Mhm.
And then the year after that, I brought home another two or three that were like this big. And then we've had two suckers since then. So we've got like one, two, three, four. I think we have six total. And they're
Awesome.
the blue Java bananas.
Oh, cool. That's
Yeah,
really rad.
I really wish we could get them to flower and fruit, but I don't know.
Those are
That's why
like
I'm
the
keeping them alive, because I want them to
yeah.
fruit flower.
Those the blue ones that are supposed to taste like vanilla ice cream or whatever. The
Yeah.
yeah. Oh my gosh, that's definitely on my list. I'll come visit if you get it fruit, because I feel like that's like tropical Eastern Asia is where the center of origin of those guys are.
Yeah,
It's
I don't
awesome.
actually
We
know, but I do know that they're a cold cold hardier. Like
Okay.
definitely couldn't leave them out all winter, but they can tolerate temperatures lower than other bananas. I have heard that you can overwinter them in Jersey. Our neighbor around the corner, they're from California and Hawaii, and he has banana trees in his front yard that he cuts all the way down and then mulches over, and then they
Yep.
grow back in the spring. I'm too scared. I feel like how can a plant bounce back from that?
You
I just
know, yeah. I cut ours back and mulched it really good with a bunch of compost and its own leaves, and I made its own like little leaf litter. Maybe this is more torture. I'm sorry, plants, but I tried to keep some familiar, plant cells around it and whatnot.
So you super quickly mentioned PGRs, and I was talking to, have you heard of a book called Yield Science?
I've heard of it, but I haven't read it yet.
Super, like, okay, so I met the dudes who wrote it. On the book, there's no author. They did not put their names because they
Cool.
wanted the book to speak for itself. They didn't want people to judge the book based on who wrote it. And which I thought was just brilliant. So I met the authors like completely the wrong way around. Had no idea who they were. Was at a conference, saw the book, opened it up, and I was like, "This book is amazing. I have to buy it." And the guy standing next to me was like, "Oh, do you want to buy it?" And I was like, "Yeah." He's like, "Oh, so my friend Matthew and I wrote this book." And I was like, "What?" So Brian is a former, what do you call it, expert hockey player? What do you call it? Yeah. Anyway, hockey
Hockey
player.
player, athlete.
Yeah, yeah, yeah. Like, but like a professional one.
Right
And
on.
so he did not look like, your cultivation like type dude. But I bought the book. It comes with an ebook. It came with like a really cool metal, actually two really cool metal bookmarks. But yeah, so I was talking to them about PGRs because there's a
Hmm.
lot of people who are like, PGRs are bad. And I'm like, but the plants have their own PGR, like jasmonic
Yeah.
acid and salicylic acid and all those are plant growth regulators. The plant will not grow without those, right? Like
Yeah.
auxins and other words that are sciencey. And so, but then there's like, and you can apply those natural PGRs to your plant to help it do whatever you want it to do,
Mhm.
including even reverse sexing,
Right.
right? But then I feel like the way that the industry talks about PGRs is all super negative. So I don't know. Can you like talk about that?
Yeah, I mean, I can definitely talk a little bit. I think, there's the natural PGRs that you can get from different sources. Like, I mean, if you make your own compost teas, there's definitely some plant growth regulating associated things shed into that. I also know that for like air plants and pineapples and bromeliads and stuff, ethylene, while we think of that hormone can be bad and cause ripening and plant senescence and things like that, it can also induce bromeliads and pineapples to flower. So if you have like an air plant or even a pineapple plant, because people have those now, you can cut a banana or just leave your bananas underneath your air plant, and the ethylene produced from the banana, the way you would kind of help ripen a tomato, can induce, flowering for other structures and stuff like that too. So that's like more of like the natural way to get the plant growth regulators on your house plants or your livelihood, your stock. And then I think about more chemical synthesized PGRs, which different strokes for different folks, right? I mean, if you have a bunch of clones that are ready to be transplanted, but you're still finishing out a different crop or you don't have the space, things like anti-gibberellins can really keep those short and stout and from kind of getting leggy, and then with auxins for rooting hormones and stuff. I can't really speak too much to like how people will spray different types of PGRs on like, buds and flowers. And I know that that's one thing that the industry gets a lot of, different schools of thought around if PGRs are good or bad for THC and CBD synthesis and all the cannabinoid syntheses pathways. I mean, yeah, it's kind of like what we were talking about earlier, a different form of inducing a response without actually the host making its own decision, right? So like you're spraying
Mhm.
something to make the plant make 20, 30% THC, or upregulating your natural THC production from maybe 20% to 28 or 26% or something, which is just phenomenal by itself. A plant being 30% bioactive compounds, just a quick aside from a microbiologist and not a plant
Right, right.
geneticist, really. Yeah. But yeah, just the way that you're kind of inducing those responses without the host making those decisions. It's kind of like a coercion to say, hey, I need this outcome, or I want this to be more purple, or I want you to not stretch out right now. I think there's ups and downs to it. Yeah. Not too familiar with what is used in the reproductive stages of cultivation when
Like
it comes
use
to BPRs.
oh, and just in the flowering stage. Do they
Yeah.
spray it on, like if you wanted to hold your clones sort of in a holding pattern while
Mhm.
you wait. Do you apply that stuff to the roots or do you do a foliar application?
I think things like anti-gibberellins, keeping those inner nodes from stretching out, basically would be sprayed foliarly and just kind of received through the leaf and the plant tissue. Obviously, auxins are going to be most supportive in the root zone, but I think the whole source sink or like xylem and phloem relationship from the plant vascular system is they can talk.
Because I don't like the plant physiology part, and then I do know some folks who that like their research was micro or muscular, or what is it, arbuscular,
Mycorrhizae, like
yeah,
arbuscular. Yeah, yeah.
and tripartite interactions, and
Hmm.
like I would listen to them talk and I'm just like, whoo.
Right on. Yeah.
Good or bad or neither. I don't know
Yeah. I
why
mean,
I
I think,
think of things in terms of good or bad, like when it's really maybe not even that.
yeah. I mean, I think like, mycorrhizae and different products like trichoderma or glomus fungi, I guess different genera of fungi that are trichoderma genus or glomus genus fungi, they're definitely just helping fill up that table that is the root zone with empty seats on it. Most of my research is in plant bacterial interactions. I'm a big mycologist, on the side, fungi. I love to go foraging and use mushrooms for food and medicine and things like that. But yeah, I mean, when you're applying like I think it's VAM, vesicular or buscular mycorrhizae fungi, and they're
Hmm.
called that because they make these giant spores that you can see with the naked eye.
Oh,
They'll germinate and then colonize the root tissue, and they can also offer benefits like phosphorus solubilization and again filling those seats up at the table. It's just more, they can also offer as like a extension of the root zone to help with, I mean, it's less of an issue for container growers, but an extension of the roots for things like even water uptake. They can act like a straw extension and help the roots maintain moisture and whatnot. So yeah, there's the main stuff you'll see in products from what I'm familiar with would be like trichoderma fungi, which are everything from like disease control to phosphorus uptake to just kind of root health in general. And then there's more things that are like host specific that you can't really grow in a petri dish, like in the glomus genus. I think a lot of peat moss, vermiculite, perlite substrates can have these glomus genus spores added to them, but it's pretty cool the way they produce them for substrate production. They'll just have a bunch of containers of things like trees or whatever the mushroom or the fungus's favorite host is, and then just kind of flush the pots and collect all the spores, and then throw those spores into a bag of soil for them to kind of just be dormant like seeds and go to sleep until they touch plant roots or moisture when they get to your grow. Unculturable fungi, right? It's like stuff that only lives in the plant root tissue and it can't really grow in a plate, but definitely there's a need for it for the host because they found it there, stuff like that.
that's, yeah, super interesting. And it's just so interesting to think about like what's going on underneath the surface. Because we can see like a lot of stuff above the surface, and I mean, we know that, there's yeast and mold, spores everywhere, but like I feel like underneath the soil, it's like a whole different world of stuff going on, including, plants above ground are communicating chemically, with not with each other, but also with insects and with other, birds, even, like, oh, I'm
Hmm.
getting munched on. I'm going to release this chemical that will attract a bird that will then eat that thing that's eating me, which I think is
Totally.
super cool. But like under the surface, plants communicate with each other via roots, exudates potentially, and
Yep,
then
absolutely.
also like these other microbes and organisms that are also sending messages, in this network of stuff underground.
Yeah, I mean it's one of my favorite, obviously places to look, right, as a soil microbiologist. And one of my favorite soil scientists, Dr. Elaine Ingham from the Soil Food Web Institute, she's quoted saying soil microbiome is like Times Square on New Year's Eve all the time. Just the amount of interactions and conversations and metabolites and traffic and all that stuff that's happening down there. We talk about the plant root exudates and how plants communicate with each other with these volatile compounds or communicate with insects or birds to help defend themselves from attacks of caterpillars and whatnot. One project that we were working on at Impello for a while that got just paused for a little bit of time, but it was what I presented on at Emerald Conference in 2023. It was how we see really significantly different bacterial communities in the cannabis and hemp for what that's worth, like cannabis sativa, both like medicinal crops, and fiber hemp and oilseed hemp. we see that they have significantly different microbial communities in their root zones when they're in veg, when they're switching from veg to flowering, and when they're in the flowering stages, right? There's a body of research talking about how the way plants build up their rhizosphere microbiome or their root zone communities changes over developmental periods. So they will kind of recruit or scream out for stuff into the void that helps with things like nitrogen uptake during the vegetative growth stages, and then even things like phosphorus and potassium solubilization or decomposition to help it be uptake by the roots during the flowering stages when the host is like most interested in uptaking those nutrients, right, to support its own growth and development. I mean, there's a review out there called like Plants Put Out a Cry for Help, talking about all the different kind of root exudate compounds that shift over growth and development or even in response to stress. But yeah, I mean, that's one thing we love to work on, or I'm really excited to work on, is like causing these stresses, things from a caterpillar bite to drought to salty soil, and then seeing how, when given like a big selection pool, right? If you just have a drought in a sterile container like cocoa or rock wool or near low microbial biomass system, there's not really anybody to hear the plant calling for help. But when you can give a plant a really wide selection pool so it can cast a wide net, seeing what these microbes, how they respond to different stresses, and then you see a few, 10 out of 50 plants do a lot better, and you see that the microbes are in those root zones. That's how we kind of help, bioprospect these naturally occurring interactions and see if we can provide them to growers if they're not too hard to culture from a petri dish to a big fermentation brewing vessel and then back into the bottle.
That do other plants okay, because so I know cannabis is the plant that I've studied the most in terms of its life cycle and whatnot. But are there other plants, like other crop plants or just regular plants that we don't use as crops that also do the same thing in veg versus flower or winter versus summer or spring or yeah?
Like I think there's even like day length dependent like bush beans, right? So the beans won't start making flowers until the lights get shorter and the
Mhm.
photo period development stuff. I know that like we always think about things like lettuce and cabbages as just we eat the green parts and we only let the vegetative stages grow. But if you've ever, it got too hot and you've seen your lettuce bolt or your spinach or cabbage or something bolt, you see the seed stalk come up. Yeah, it tastes bitter because its chemistry is changing, partly from the way its root exudates are changing, I'm sure too. But yeah, I mean, the photo period development plants, I think cannabis is definitely the most famous one when it comes to that. But a lot of different things you'll see them change from vegetative to reproductive growth when the lights get shorter after the solstice and whatnot.
So Colin Bell, who I've known, I mean, I met him at my very, very first conference, presenting my very, very, very first bits of, data that I have found
Yeah.
doing cannabis. And he had just started Mammoth Microbes. And he ran up to me and he's like, "Oh, your research is so interesting. I'm Colin Bell. I'm starting this company, this microbes company." And he gave me his card and he talked to Mitt and they talked nerd for a while because Mitt's a plant guy and I was just, like I'm a geneticist. Like I've never really been a plant person because genetics is
Mhm.
genetics, right? But obviously I'm working with plants, so I know a little bit about plants, but I'm not really a plant person. But I
Yeah.
remember Colin came on the Dank Hour too, and this was back when we were doing Clubhouse stuff, so it wasn't like a video chat or anything like that. And he was talking about like, and I think Tess was involved in this conversation too about figuring out what microbes are beneficial like in the desert versus in a wet situation so that you can create a microbial community that will best benefit your situation or what your plants are going through.
Yeah, absolutely. Well, I mean, Colin's a great guy. I sat on a panel with him last year or 2 years ago at MJ Unpacked talking about the use of microbes in, controlled environmental cannabis operations. So it was the first time we met, but I know he's a neighbor up here in Fort Collins in Colorado too.
Hmm.
And yeah, Mammoth was definitely the first micro product I saw when I was growing cannabis back in college and whatnot.
That must
But
have been
yeah,
an MJ Unpacked in St.
it
Louis.
was in St. Louis. Yeah, I think it was 24. That was the first time
Yep.
I got to go to that show. It was a fun one.
I love
But yeah,
MJ
I mean,
Unpacked.
yeah, my favorite term, I think, and I think I identify myself like this on LinkedIn, is like as a bioprospecting enthusiast. So instead of prospecting for gold or different elements and stuff, bioprospecting, seeing what natural, biological materials or living organisms respond to different stresses, or seeing the environments that they exist in and just they're happy, right? You're walking through the desert and you see a really, really established cactus. It's because that host is kind of equipped to live in its environment through millions of strategies, but some of that could also look like the root zone microbes or the foliage microbes too. I know there's some kind of radical bioprospectors, and I say that in the best way, where they'll go out on their hikes and whatnot and see a really super established plant stand of like grass or mullein in like a burnt forest out here in Colorado, collect a little bit of that soil in that area and then bring it home and try to cultivate those microbes in their garden. I think it's a super cool take, you know, and the way we kind of work on this stuff at Impello is we'll do really designed experiments in soils with a lot of, you know, alpha diversity, showing really balanced different species, and you know, giving the plants a wide selection pool of microbes to recruit from, and then, you know, stop giving them water for a handful of days and implement a drought, rehydrate, see what plants bounce back the fastest or which ones didn't die, and then, you know, we'll use DNA sequencing. We have an Oxford nanopore to look at bacterial communities, and then basically just look at the control plants versus the ones that were under the stress but did okay, and try to find beneficial microbes and see if those things are feasible for product development. But also, it's really cool research, and like everything from buying a bottle of bacteria soup on the shelf to going out to the forest and collecting some, you know, healthy soil debris, or even your own compost pile. Compost pile is definitely a type of bioprospecting too. But yeah, like the desert ecosystems versus the wetlands. I can't remember this gentleman's name, but there's a guy on Leighton Morrison's podcast, The Soil Matters, and he used to, like, he was one of the more kind of collector bioprospectors. He'd go for different hikes and see plants doing well, and then just try to have like different raised beds in his backyard, and like, this is my drop bed, this is my flooded soil bed, and kind of growing different plants in there with these microbes and trying to maintain those conditions, and seeing it's kind of bringing natural environments back to your own cultivation room. I think is super interesting, and where we can learn a lot from the natural relationships that are already happening.
The idea of going and like foraging for a microbe community. And I don't know why it popped into my head, but like fecal transplants, you know, like
It's really similar, yeah.
people who are not healthy taking, you know, fecal matter, and you know, you don't have to eat a pile of poop, but they put it in,
Right.
try it out, put it in capsules, and then give it to somebody who's super healthy, to
Hmm.
somebody who's not doing, you know, doesn't have a great gut microbiome and isn't doing very well, and basically, you know, supplementing a gut microbiome. So that makes a lot of sense when we're, you know, thinking about what you said before about the root zone is like the plant's gut microbiome.
Yeah, I mean that's like a perfect linear analogy. Like I think a fecal transplant is as gross and as out of the cultural norms. You know, it's not something we're all talking about every single day, right? Actually, I know that the best way to get those bacteria to colonize is actually a tube through the nose down to the small intestine instead of eating the poop pills, right? And
Hmm.
yeah, it's getting gross. But yeah, I mean, actually, I have a lot of gut microbiome scientist friends, and when they do their DNA extractions from those fecal samples, they use the same kind of kit chemistry as we do for soil DNA extractions, because the matrix is really similar, as you know, dirty, for lack of a better way to put it. I think that like the fecal transplant, you know, I'm gonna leave my house for a week, and you know, I wish it was this clean of a logic, but I'm gonna go collect a bunch of soil from the desert and then compost or till in my desert soil with my garden out back, and then it'll survive drought a little bit longer while I'm on a trip or something like that. I think
Hmm.
it's not as black and white as that, but you can build up those community resiliences, and you know, like reapplying and kind of stressing your plants out and testing them out. You know, if you have the ability to do research, a lot of people just grow for livelihood and whatnot.
Another thing I am not super familiar with, but I have had some, you know, we've talked to people on the Dank Hour about it, and I've talked to Steve from Potent Ponics about this. Is the we were talking about compost teas, right? Making you can buy compost teas, you can buy, you know, like all the ingredients. But Steve was talking about like make your own, but like, and I guess it comes back to part of like the idea of Korean natural farming, right?
Hmm.
That you can make your own, I don't know, mash juice stuff where you like put out rice or something and basically like just leave it out, and microbes will come to it and like start establishing it, and then you can use that to make your own mixes instead of purchasing something off the shelf, which may or may not be conducive to your particular area, especially when you're growing outdoors or in like hoop houses or something.
Yeah, I think that kind of lumps back into the radical bioprospecting kind of, you know, like practice. That person that whose name I'm blanking on, they would use rice cakes. I think like a little bit of sweetener, but mostly just the carbon and whatnot, and bury them in the forest or somewhere they saw a plant or an ecosystem that looked really interesting. Leave it for like a handful of days to a week, and then come back and collect those rice patties or the rice cakes and
Hmm.
break them up over their soil, and then yeah, use that as like a way to inoculate. It's everything from like growing mushrooms or like a CO2 bag, right? Like you see all that mycelium in the bag. go collect it and spread it around. Yeah, but like we going back to like cakes and cookies, right? Like the root exudate, the carbon compounds that the plants are putting out, that carbon from the starch and the sugar from, the glue that makes it like a little pastry. Like that is a great way to kind of, expedite the recruiting of microbes and kind of concentrate them onto a space where you can, go apply them or brew them with compost tea. Yeah, compost tea is really interesting too, because I think depending on the type of input you're brewing or, aerating, right? You just kind of bubble it with an air stone for 24
Hmm.
to 72 to however many hours your heart
Hmm.
desires. You can kind of start to select for communities that really like humic acid, like aerated humic acids, right? So there's a sweet spot that I think is still being looked at in the literature and by different scientists and whatnot. Like if the line graph is like 0 hours to like 96 hours here, you start to see decline after a while. So much so that people are even like boiling or autoclaving their brews to kill all the microbes, but keep all those fulvic and humic acids and all those other organic materials that are released in there. So after you let it sit for a while, some of the microbes start to chew up the stuff that your plants want to. Plant microbe
Mm.
competition can be accidentally, ongoing in
Hmm.
your compost tea bucket or any type of composting pile.
And I can't remember, we were using compost tea for our grow for a while, and I can't remember what all we put. I know it was worm castings and molasses, which is
Hmm.
this cakes and cookies,
There we
right?
go. I've done it like that before, where I just use vermicompost in like a muslin bag or
Hmm.
like a oat milk bag, and add a little bit of sugar to feed the microbes. And yeah, just trying to dial in the time, I think, is like the hardest part. And it's without a, you can't always sequence your pile and have those results on what communities
Can
are
you do it
happening
wrong? Like, is
immediately.
there a possibility to like create or recruit microbes that you don't want?
Yeah, I would say, there's some people that are really like when you see like a dying or sick plant, or even just you've overplanted something in a garden or in your own grow where like you actually need different space or something like that, and you start to cut things. I've heard some folks say that they're really averse to putting like chopped tomato leaves or leaving any kind of type of plant debris around their healthy like tomato plants, let's just use a hypothetical, because things that are going to break down that non-living tissue anymore might also be like saprophobes, right? So where they can break down dead tissue or living tissue and then jump over to your plant host. So I think, yeah, like they say, if you can't get a compost pile hot enough, it's hard to I mean, worm compost versus like microbial compost are different stories, but for the microbial side, if it doesn't get hot enough and you have like plant viruses in there, they're not going to get denatured, and you're going to treat your garden or your grow with viral infused material. So yeah, there's definitely ways you can kind of grow or select for microbes you don't want in your system. Unfinished compost, things that smell really bad, like the even before we're getting to like the tea and the brewing stages, like I think the ideal carbon to nitrogen ratio is like 30 to 1. So if you have a compost pile in your backyard and it smells really bad, you probably have a lot more nitrogen than carbon. So there's like a lot more things that can break down nitrogen that are anaerobic and they stink, right? And then if you have a lot more carbon than nitrogen and you're not seeing, you're seeing the same tomato in your pile every day for 2 weeks, there's not enough organic material or carbon to help the microbes break things down. So like that's why you want to balance those ratios before you even get to like the decomposition process, before you even get to the brewing process, right?
I've never composted anything because I just feel like I would screw it up.
It's one of my favorite things to do. I helped like with a composting club when I was at CSU called the Zero Waste Team, and yeah, I think it's super rad.
That's cool. Yeah, my dad had a compost when I was growing up. My dad used to, we had a greenhouse and an outside garden, grow all of our vegetables, and we had chickens and stuff like that. And we had a container on the bench that all the food scraps would go in. But that was all for, I don't think we had separate ones for compost versus chickens. I think the
Hmm.
compost was mainly for like the vegetable matter from the garden, and then the scrap bucket in the kitchen was for the chickens.
Awesome. Yeah, chickens are totally on my list because I think my friends who have them, they're like pre-composters, and chicken manure is a great fertilizer too, as long as
That's
you
what I've heard, and I
yeah.
was too little to like really understand what my dad was doing all the time. But the garden was great, and the chickens were great, and I had to go get the eggs, which I hated because then you have to walk through chicken poop.
And the moms are gonna peck at you if you're getting underneath them and
Yeah, they can
and
get
stuff. Yeah.
yeah, yeah. But I would really like to have chickens again, and I wish I was talented enough to grow my own garden, but I'm just not. I don't want to say I'm not talented enough. Lazy, I think is the word. I
Respect.
just not attentive
Yeah,
enough.
yeah, yeah. One of my favorite memes is like, we put $30 into our garden this year to get one tomato and save 30 cents or like something. Like there's definitely an ROI, return of investment, like that is not always worth it. One of my favorite topics in cannabis cultivation is like the myth of sterile cultivation systems. And I think, people are starting to kind of move away from that. But when you kind of dose things like peroxides and ozones and all that kind of nanobubbles, I mean, bubble aeration's great, but if you're doing like chemical additions, I mean, yeah, there's different schools of thought. They're definitely great curative situations if you kind of notice a biofilm outbreak or like a disease outbreak in some of your environments, especially hydroponic systems, right? But yeah, I mean, I think 5 years ago we would see a lot of people metering things like chlorine or hydrogen peroxide or peroxyacetic acid into their systems all the time to maintain a super clean line, sterile system, all that kind of I think maybe science that is well understood when it comes to, if you don't let anything grow, then there can't be disease, right? But I think an even better way to kind of frame that lens is if you populate, get some good guys sitting at that food table that is the plant rhizosphere, even just one inoculation during the cloning stages, not applying it every week or every 2 weeks or something like that, is a really good way to prevent diseases and biofilms and trying to, maintain like a balance of
Mhm.
different types of microbes in your system, things like the mycorrhizae and the vesicular arbuscular mycorrhizae, the VAM, you know, as everybody talks when it comes to cannabis genetics or microbiomes or team workplaces, right? Diversity is always really good. So pumping like chlorine into your system all the time, you'll eventually get things that can tolerate that level of chlorine, and maybe they're not great for your plant host versus pumping like compost teas or organic matters or biologicals into a system is a great way to kind of prevent disease outbreak, and it's a kind of like maintaining a little like water ecosystem with your plant roots being like the main food sources for stuff in there. I don't know if you hear any more about people talking about sterile cultivation systems too much. That was one thing we used to really, a lot of our customers would talk to, and a lot of people that I would collaborate with in the cannabis spaces would be like, "No, we don't want microbes because they're on a sterile system." It's like, did you
Yeah.
go into your
Yeah.
lab with a wetsuit on or a biosafety level 4 suit? Because if you didn't,
God, but yeah, diversity is like key. And you said, maintaining a balance. And I feel like we all know that monocultures are bad. You were talking about companion planting, right? To introduce that introduces diversity. And we're as humans, we do so many monocultures and we're so stupid. We don't learn anything, right? We almost lost the papayas. Like when there's no diversity, there's no ability to react. You got one disease that comes in and now you wipe everything out. So same thing in cannabis. If you're maintaining a sterile environment, you get one bad thing that sneaks in there, your whole crop is yeah. And I that whole idea of taking up the seats at the table, if you've got all the good guys hanging out, there's no room for the bad guys to move in. And whether that be
Yeah,
companion plants that are attracting the aphids that could otherwise attack your plants, or, whatever the case may be. I think
right.
diversity is something that we don't think enough about, and we don't put enough importance in it.
I mean, especially like on the microbiome, and we can't know how to correct or help support what we can't see, and it's really easy to just kind of not think about the below ground diversity and the microbes that happen there.
Mhm.
I mean, just like eating like a super colorful plate of carrots and greens and potatoes and meats and whatever your diet is, people say colorful plate. There's a lot of different microbes in your gut that can break down different things in carrots versus collard greens versus broccolis and meats. If you only fertilize with the same salt all the time and there's no organic material in there, you're just kind of procuring an environment that doesn't really encourage different diversity, different foods. Your plants can only pump so many different root exudates into the dining table that is their root zone.
Mhm.
That's why I think balancing conventional nutrition with organic or biostimulant based nutrition is a really good way to kind of encourage that diversity that we're talking about.
Mhm. For the like industry, like the commercial grows. I mean, I feel like they're never going to steer towards the full on hippie organic compost teas kind of situation just because it seems like it might be a little bit difficult to control
Yeah, definitely.
and could potentially introduce problems. But I also think that the idea of having a sterile cultivation is also not good. So what do you, I mean, you work with people in the industry, and do you think that the attitudes are kind of changing then towards like being able to utilize microbes and other things in a sensible manner with research behind it? Do you think that the attitude is changing from the sterile idea to more of like a more responsible cultivation thing,
Yeah,
whatever
like
that word
mimicking
is?
like a natural, yeah, like trying to mimic something more, natural or something that your plant would experience in the
Hmm.
outdoors, right? Which is not always easy to control either, or definitely impossible to control things like weather and insects and all that.
Yeah.
I would say, yeah. I mean, I've been working, in this space since 2020. Most of, I think, 2020 to 2022 or so was really trying to convince people that biology and beneficials can tolerate your, high potassium nitrate, salt or some kind of solution in there. But, there's the difference between like like biological nutrition and conventional nutrition for all types of growing systems, not just cannabis, but also commercial agriculture production. I think it benefits from a little bit of everything. I think the person that's only applying salts and sterilizing agents is going to miss a lot of, microbe or interaction based product benefits, right? Like things like this really high quality THC or really fat trichomes and terpenes that mature maybe a little bit early because of that induced systemic resistance or systemic acquired resistance that can like help your plants mature maybe a little faster, right? Yeah, I do think it's shifting. I'm not going to say like it's perfectly balanced. I think there's definitely a lot of older head kind of growers that think they maintain sterile systems and whatnot, but I would say the reception to things like biologicals or even organic inputs, just the integration, right? They're all tools in a tool belt. And if you have a diverse tool belt with a lot of different things on there, like again, going back to diversity, people are starting to see the real time benefits, and they just don't need people like me chirping at them saying biologicals, apply them to your root zones early, early, early. But
Hmm.
trialing things out. One thing that we like to do, and this was a lot more popular 5 years ago than it is now, is we have a bacillus product with 3 and 4 species of bacillus bacteria and pentobacillus bacteria in them, and we developed a qPCR assay, quantitative polymerase chain reaction, where you can send us a little bit of your root tissue or plant tissue, and I'll do the DNA extraction at the Impello lab, and we'll run a qPCR assay to tell you what those populations of the species in our product are. So everything from microbiomes being a huge part of maybe the scientific jargon and zeitgeist, and there's documentaries on that stuff now on Netflix that are really, not, it's like exited like the scientific gatekeeping with the big jargon and all that stuff, and people are starting to understand it a lot better because probiotics are on every shelf at the grocery store.
Hmm.
But yeah, we're just trying to show people that like, yeah, you're spending money on a product, you're applying it at this rate. Here's the level of the bacteria in the bottle. Here's the level of the bacteria in your root zone, because they want to be on the root zone a lot more than they want to be in the bottle. So not only are they colonizing or populating the rhizosphere, but they're growing and living in their favorite environment. So yeah, it's been interesting to see the shift, and I hope as the time goes on, we'll start to see even a greater adoption of microbial products, or even just the more trickling introduction of things away from purely conventional. I think there's always a reason to do both.
Yeah. I feel like people are, yeah, becoming more aware of the products. There's soils that you can get with things like guano already mixed in there.
Yeah.
I've also heard people say that guano is not great because it's too high in nitrogen. I don't know if that's true or not. Fish shit is a
Yep.
product out there. I've
Totally.
used it. It used to be there was you could buy fish meal, right? Which
Hmm.
is just the bones and the blood and stuff, whatever. I don't know what it is. But then fish shit came along, which is a similar product that it's one of the things like every now and then I'll be like, oh, I should feed my plants. And I'll mix up some, like Big Bloom or something. I don't even pay
Totally.
attention
Yeah.
really. And some fish shit and then just feed the plants.
Yeah.
They seem to
I
do
mean,
fine.
like you said, like it's really hard to scale up to commercial production these kind of hippie, organic, like almost homeopathic practices like fish shit, right? And bacteria or compost teas and whatnot. And I think that's the reason people like Impello and fish shit and Mammoth at the time they were around, and all those other companies start to kind of they see a need for the metering of the conventional with the biological, or the chemical
Hmm.
to the organic and whatnot. But I think if you're doing only one of those two things, you might be barking up the wrong tree depending on the size of your operation. We have I think homeopathy is a great way to take medicine and do medicinal practices and whatnot. I'm a big wildcrafter myself, plants and mushroom medicine and stuff. But I think like if I had a pretty severe diagnosis, I'd probably also seek a physician's contact and see what I could do to complement whatever they're giving me with whatever's I can find in the woods and cook up in my kitchen or the
Yeah,
lab. Yeah,
yeah, for sure. Like if you've got cancer or something, like do what your doctor tells you, but also maybe adjust your diet so that you're, got the best nutrition that is not conducive to growing the cancer or something.
exactly. Yeah, and I think a similar like school of thought can be practiced in a hydroponic or any type of production cultivation system, from strawberries to cannabis to flowers, like petunias, right? Or like something like an ornamental plant. Yeah.
Petunia.
Yeah.
Okay, so these products that you can buy on the shelf now, they do come with instructions and everything. Like you can buy things in buckets, 5-gallon buckets for a big scale operation where it tells you like, if it's in this week of, veg or this week of flour, like this is your product and this is your measurements to make up this many gallons. They come with pretty good instructions these days. But these things have to have a shelf life, right? Because they're full of things that are alive and
Hmm.
they don't stick around forever, right? So
Yeah.
how long do these things last on the shelf or in the fridge or wherever you're supposed to store them?
It depends on who you're looking at or what types of bacteria or fungi or even just general microbes are in there. There's like prebiotics, probiotics, and postbiotics, both for the plant industry and the gut, the human guts and the mammalian guts, right? So prebiotics, they're like fibers for the gut or organic inputs and like algae and compost is like compost organic materials that microbes can access. So prebiotics, they feed the biotics, the microbes. Then there's
Oh.
probiotics, which are the living organisms themselves. Things like you'll see bacillus products in a bottle, or trichoderma products in a bottle, or pseudomonas and those different types of bacteria genuses or genera. Then there's postbiotics, and those are things like the enzyme products that you can see, or peptides and stuff like that are made by the microbes but not living, right? They're just the things the microbes produce. So those are like three really big umbrella terminologies for what you're looking at. And the former and the latter, the prebiotics and the postbiotics, are normally just kind of like organic materials or microbially produced products, everything from like insulin to auxins, right? They can all be made in a bioreactor and then purified and sold as a microbial product or a postbiotic. But when we get to the
Mhm.
probiotics, to get back to your question, like the shelf life of these things, it really depends on what you're looking at. And I can speak more about bacteria than fungi, but fungi is kind of quick. A lot of those different organisms, especially the plant benefiting ones, form like from one to like seven different types of spores. And all those have different environmental tolerances. They can survive freezing and thawing and high heat and super cold temperatures, low oxygen systems like the bottom of a hydroponic like solution that's not being. But yeah, back on the bacteria side, there's two major classes of bacteria. They're called gram-positive and gram-negative bacteria. Gram-positive are ones, and that is just basically how thick and how tough is the cell wall of the actual cell of the bacteria.
Mhm.
Gram-positive ones are a lot more tough, and a lot of those, depending on the like taxonomy that they're assigned to, they can form things like spores, which are basically bacterial seeds. Like a really equivalent would be like if a bacteria can make a seed, like a plant would make a seed. It's going to be
Mhm.
this dormant structure that's going to hang out in a bottle, in a dry soil, in an ice cap until the environment triggers its germination. So like bacillus, I can speak to because I'm pretty familiar with those species. They have like 3 to 6, really common germination receptors that are triggered in agriculture and controlled environmental agriculture and horticulture. They can be things like hydration to plant root exudates or contact on the roots. But those spores can survive like up to 500 years, depending on the environment. There's a lab that has a time capsule of bacillus spores, and they started a project, I think 50 years ago now. And every 25 years, whichever grad students are in that lab dig up this time capsule and then measure how many spores are still around. And I think in the first 75 years, they haven't seen any loss of that. So spore forming organisms are super resilient to shelf lives, but all companies will either put like a year, 18 months, 2 years, or sometimes even a 3 or 6 months shelf life on there.
Mhm.
So, definitely important to use that stuff up. But we've had people say, hey, I just found this bottle in the back of my closet when I was moving my house. It was from 2021. Is it still good? And we'll either ask them to send us a sample or use our internal data to say, well, this is bacillus, so it's probably all spores. Make sure you shake it really good and there's nothing else growing in there. But yeah, we have data for 5 years old, a hot car trunk, the bacteria is still up over the label claims. But we have to be reasonable when you start to sell stuff when it comes to like a registration and like regulatory space where you got to put a
Hmm.
shelf life that's reasonable on there for the states to approve your products to be sold. And that's less of my wheelhouse, that kind of business and regulatory side. But like with the, going back, gram positive, those guys are pretty good at being resilient and staying around for a long time. Gram negative bacteria, on the other hand, they have thinner cell walls that are made of less resilient stuff. Peptidoglycan is like the chemical it is, but they're thinner, and they, if people like to joke in the fermentation world that if you look at a gram negative bacterium the wrong way, it'll die. So, like, yeah, I've there's a lot of different, prebiotics though that other fermentation scientists and other companies are putting in with their gram negative bottles to slowly kind of maintain a food production or a food source for these bacteria to kind of like be like a everlasting gobstopper, like still alive, although maybe declining in viability over time, depending on what the environment is. A lot of people will say keep the bottle out of direct sunlight in a cool, dry place,
Hmm.
because if it's really hot, the microbes will grow. They like temperature changes. There's a reason
Hmm.
we grow bacteria at 37 C or 98.6 Celsius.
Right.
They like it warm, like inside us. But yeah, another really cool aspect I've seen other companies do for these gram negative bacteria is kind of like sell you a bag of fermentation media and then sell you a little spore powder that is like freeze dried cells of the bacteria that you're buying. And then when you're
Hmm.
ready to use it, you mix A with B on the farm or in the facility and shake it up, and the bag will start to swell. It's basically like a bag of wine, like Frenzia or something, and it'll start to kind of ferment, and it's got a little doser on it, and you can have the microbes in their most active growth stages because they're active, they were just waking up, like kind of like when you get sea monkeys as a kid and you put the powder in there and you watch for 3 days until they start to populate. Really similar stuff. You grow the bacteria at your farm and then inoculate it onto the root zone there. So
Yeah, that's
there's
cool.
solutions for these really hard to survive microbes, and some of the stuff we're working on is gram negative, and we've debated a lot of these different solutions. It's right
Yeah,
now.
I mean, it seems like it's a like a puzzle on trying to figure out what you're gonna put in, and what you're gonna, I don't know, not avoid, but like what's your solution to your problem?
It's a biological process. Stuff will wake up sometimes, and like I've seen other companies' bottles kind of like bloat and explode from different, things like that. We had a really good food source product made out of corn, the corn wet milling process that we are starting to bring back into the market, I hope this year. It's a great microbial food source, and it's really hard to sterilize stuff at 1,000 liter scale without the proper equipment. So there were times where I would bring some home to run in my garden and forget about, and then this bottle's expanded and I open it up. It's like fermenting kombucha or kefir or something at the house. It off gases. So, there's a reason a lot of the microbial products people see in the market are really similar species, a lot of bacillus, a lot of trichoderma and mycorrhizae and stuff, because those are the ones we figured out are
They're okay.
practical in a commercial
Yeah.
industry. Yeah, they can live for a while. They're not going to blow up or change smelly, get smelly halfway through. Or if they are smelly, that probably means they're working really well. But if the smell changes as you just open and close the bottle a bunch of times, that's not good for anybody. Things like that.
Cool. Well, microbes are pretty weird.
Totally.
Do you do a lot of like microscope work, or are you kind of past that? Yeah, still?
Yeah, I still do a decent amount of microscope work. Anytime we're working with something new, we want to understand it and try to identify it in-house without spending all the money to do DNA stuff, which we can do in-house, but depending on the organism, we can outsource too. But like, yeah, you can, put some different dyes and stains on the microbes and tell you if they're gram positive or gram negative, and then that cuts out a whole, tree of bacterial taxonomy saying, well, if it's gram positive, there's no way it could be any of these things. So a lot of identification based stuff. If you're a customer and you're dealing with maybe a water mold like Pythium or Phytophthora or something, it's of interest to us to get that in our strain bank collection to kind of like see how that interacts with different solutions we might be working on to release down the road. But my favorite thing to do, and what I most of my graduate work was, is like sequencing data. So like we'll design an experiment where we'll have these 20 cannabis plants be under drought and these 20 not be under drought,
Hmm.
give them some kind of natural selection from like an organic soil or a compost or something, see which ones do good, and then like kind of analyze the differences between the ones that did good under drought versus the ones that didn't do good under drought versus the control plants, and to try to see if these communities are different enough to pull them back into the petri dish, back under the microscope, and see if we can, get them in the bottle for people who might not have them in their system.
I feel like sometimes some of the stuff that I've done has worked because I didn't follow the directions.
I think, there's more than many routes to Rome, right? Many routes to Rome. There's a lot of different ways to reach the summit of a trail. And like, yeah, like you said, that doesn't mean the most commonly practiced way is the right way or the best way are going to get you the data you need.
Yeah.
And yeah, with the microbiology exposure stuff, you know, I moved to Colorado because I was working on a brassica cabbage and collard green farm in Maryland for a year or so, and I figured it'd be cool to study this stuff and come back to the farm and see what I could do. But I, you know, had bills to pay, so I got a job on campus. I didn't have a vehicle. I was biking around everywhere, and I kind of bumbled my way into a microbiology lab that needed a dishwasher, essentially. Yeah, I got my first exposure to all that stuff just kind of as a grad student aide, and really fell in love with the microbiology and, you know, petri dishes and watching fungi grow. You don't know what you're missing until you get exposed to it, I guess, to talk about the microbiology class that you took, and like how it's like, wow,
Hmm.
yeah, there's a whole nother world down there. I recently got a pair of binoculars from the thrift store I found, pretty nice one, and I swear this will get back to microbiology. And yeah, I feel like I, you know, spent so much of my career looking under microscopes. Now that I'm out on the trail, I use the binoculars, and it's like just,
Yeah.
you know, another kind of microscope, seeing the peak from across the way. It's fun.
Yeah, for sure. I met E.O. Wilson, came to our school and did a presentation. That would have been like 2000, maybe 16, 17, maybe 16, I think. And he, he's a famous naturalist
Totally.
and wrote some great books. The Half Earth Theory. If anyone's interested, check him out if you don't know who he is. But he was really old by this time, and we were showing him around our little teeny tiny herbarium at UNC, and he was telling us how he got involved in the natural world and got interested in biology. He was already interested in the natural world, but he really loved birds, like bird watching, wetlands, things like that. Him and his brother were playing, and there was an accident, and his brother damaged his eye to the point where he couldn't see, like
Mm.
he couldn't see far away, he couldn't see through binoculars. And so he turned his attention to ants. And so that's how he became involved with ants because he couldn't see. So seeing,
Yeah.
the behavior of the ants, it's almost like, a city and it's a whole ecological system. That's how he got really involved in ants and smaller organisms. And I thought that
Well,
was really
I yeah,
interesting.
I love a good origin story that kind of feels like an accidental exposure to something that like is a, you know, a love or a passion you didn't even know you had. Super, super neat.
It's weird how we end up where we are because I was never a plant person, and I've told the
Yeah,
story
how did you get
before
into
that.
plants being a geneticist? I've not heard it. I'm sure you maybe said
Well,
it on
I
the
started I
podcast
started
before,
at
but
CSU with animal anatomy and physiology, thinking I might want to be a
Mhm.
vet, animal lover, and then I I turned 21, my fifth year, got 0.0 GPA on 18 credits. And my parents were like, "We're not paying for any more school," and I was like, "That's fair
Wow.
enough." So my plan was to work and go back and finish the 12 credits I needed to graduate, but then got pregnant. So then I went and I worked in pizza for like 15 years. And then one day I was like, I
Mhm.
kind of want to go back to school. So I went back to school and UNC, I transferred all my credits to them.
Cool.
Had not passed organismal biology, couldn't pass organismal biology because of the stupid
Mm-mm.
plant part. I hated plants. Plants are stupid. And but UNC didn't have an animal anatomy and physiology emphasis. So I was like, all right, cellular and molecular biology. Maybe I go to
Got
cancer
it.
research or something like that.
Hmm.
And in order to graduate on time, I needed an upper level biology course to fulfill, and I needed my capstone credits. I needed 6 capstone credits. So the class that I wanted to get into was animal behavior, but it was full. It was over full. There was like 5 people on the wait list. There was no way I was getting in. But if I didn't
Dang.
take it, I wasn't going to graduate and have to wait another whole semester. And so there was this stupid plant taxonomy class. That was the only class that was open, and I was like, fine, I'll fucking take it. And it was Mitt's first class. He had just started at UNC. This was a brand new class, brand new professor. He was really excitable. He looked like John Cusack and just really excited about plants. And I was like, okay, well, maybe it'll just be fun. And it was pretty fun just to tell people like plant blindness is a thing. We had 100 must know plants. And then on top, we had to learn those, and they're all Colorado native plants. And then as the semester assignment, you have to go and collect 20 plants that are not on the must know list, and you have to identify them, make a herbarium, like pressing, do a label. And I was like, that, but that's like all the plants. Where are we going to find any more plants? Like, there's nothing else there's no more plants on that are not on the list. Because that's what I thought. Like, that 100 is a lot of plants. That's not a lot of plants.
Totally. Yeah. Yeah.
But I did that. And then I needed my capstone credits, and he had the plant population genetics lab, and he was working with the Denver Botanic Gardens and the Bureau of Land Management on this cactus project. And he's like, okay, so to get 6 credits of capstone, that's like 100 hours. You're gonna have to work really hard, like every day, pretty much, to get the credits. And I was like, that's cool, because I was taking one class. It was my last class that I had to take to pass. It was molecular genetics. And he's like, okay, so we've got this cactus hybridization project that you can start working on. So I was extracting DNA,
Mhm.
did not follow the directions, um, and forgot to add the R proteinase K to the extraction thingy,
Okay.
which happened to work really well. And further on down the line, somebody else started doing the cactus extraction and it wasn't working at all. And I was like, what's the difference between the two protocols? How come mine works and yours doesn't? She was adding proteinase
Uh,
K, and I was like, You don't need that." And then it worked.
cool.
I'm like, that's, like we might have never gotten any work done if I hadn't have figured that out. But so I worked on this cactus project that turned into a master's project. Then I worked at the Denver Botanic Gardens on all of their conservation genetics projects. And then
Nice.
cannabis was legalized, and I was like, hey, I want to do this. And it was like, no. And I was like, okay. And then he was like, yes. And so then I came back to his lab and started working on cactus, which I also implemented the non-proteinase K DNA extraction CTAB method because it works on getting around some of those secondary compounds, I think. And it
Mhm.
worked way better than the Kyogen kit or the regular CTAB protocol. So
Cool.
yeah, so
That's
that's how
really
I
rad.
became a
Yeah.
plant person.
I love it.
I
I love
got
a good
shoved.
bumbling story. I hope, yeah. Okay, shoved, bumbled, either way, it's super.
Yep.
Yeah, yeah.
Dragged,
I
kicking and
honestly,
screaming.
as somebody who looks at the microbiome a lot, I do look at like plant tissue, like not cactus, but we've done cannabis leaf stuff. And Kyogen actually told me that I should collect my samples on the farm in, I think, proteinase K and then like leaf discs or something. Or maybe it's like a stability buffer. I can't remember the actual reagent, but yeah.
Some sort of buffer.
But it was like to kind of, denoise the plant host, the plant cell DNA from the microbes that are inside and around it and stuff.
So the primers that I developed were species specific. So like you could sneeze into the tube and the PCR tube, and the
Would still
only
work.
thing that would amplify is the plant. So
Nice.
that's one of the bonus things doing the kind of using the kind of genetic tools that I used. They were cheaper. I didn't have any money for my project, so we needed to do something on the cheaper side. But yeah, if I was doing whole genome sequencing or something like that, like not species specific, I could have been and probably would have been amplifying all kinds of shit because so used to working with species specific things.
Totally. It's kind of cool, another nerdy thing, but like looking at the plant DNA, like or the microbes that associate with the plant DNA, when you do a 16S PCR or a
Hmm.
16S like next
Hmm.
generation sequence run, the chloroplasts from the plant cells have 16S too, and so do the mitochondria from the environment or the lab tech who did the DNA extraction. So you actually start to
Hmm.
copy and amplify the mitochondria and the chloroplast from that stuff, and you have to filter them out of the 16S data set because they just kind of, coalute and whatnot. It's like that whole two bacteria, one bacteria ate the other one, and then the symbiont stayed alive in there to make eukaryotes. No symbiont theory. This
Yeah.
came to me.
Yeah, we had problems not doing species specific stuff at the Denver Botanic Gardens because, as you can imagine, there's a whole lot of plant shit everywhere
I
flying
bet.
around, and we didn't have like a proper hood. We had like a reverse laminar flow hood, but yeah, like the contamination, they were having all kinds of contamination issues. I'm like, why aren't we developing species specific stuff so that we don't have an issue with contamination of other
Yeah.
plant stuff?
Was
You
that
know, uh,
cactus stuff at the
it
DBG or was it
was.
all the plants there?
So UNC did the cactus project, but they had a whole bunch. I think I worked on 13 other projects all in like at the same time. We had Facilia, there was a coffee project, there
Nice.
was a fungus project that I worked on. Yeah, there was like 13 different projects that I worked on, and they were all either side projects, or like the coffee one was a side project, or
Cool.
it was for land managers looking at, rare plants, sensitive species, things like that, because they want to come and drill all the oil. And if there's a rare or endangered species on that land, they can't do it. So they wanted to know, are these populations pure? Are they hybridizing with something? Can we drill here? Can we not drill here? Can we build an airport
Interesting.
over this?
Classic, man. Yeah. Was that like a Denver Botanical Garden and UNC like collaborative thing, or did you go from UNC? to DBG and you were like a researcher there or something like that? Like, what are the
So the BLM gave money to the Denver Botanic Gardens to figure out this sclerocactus project, the cactus project, and UNC picked up the bench work to do that because they didn't have a lab, a functioning lab at the Denver Botanic Gardens at the
okay?
time. So it was kind of like an outsource, like you do it, and then we'll collaborate, and then with the BLM and all of that. And then the other projects were all BLM. UNC is still working on the sclerocactus project. They've expanded the species list into, an area out of Colorado into Utah, down into New Mexico, and are looking at, because it's historically, just like cannabis, been a really convoluted taxonomical problem because
Hmm.
sclerocactus are also super plastic in their phenotype, their
Didn't
phenotypic
know that.
expression depending on what resources they have access to. And so, like, my research found that there's actually, like, they're not really hybridizing that much, like you would expect for two really closely related species that basically overlap in their geographical regions, but there are actually two, like the one species that grows in the area around Grand Junction. Those are actually two different species. Even though they look really similar, they are really, really different from each other genetically, so that we were able to name a new
Hmm.
species. And then the Utah species, there's one
Cool.
called Wetlandicus and one called Brevispinus. So Wetlandicus is like huge. It looks like a giant cactus pineapple. And Wetlandicus is this little tiny baby, cute little like half ping pong ball to half tennis ball size. Adorable. These little tiny little spikes. Wetlandicus has these great big angry spikes all over it. Those two are actually the same species, just growing in different
Wow.
habitats. One's got a lot of resources and the other grows in like dry creek beds and stuff and doesn't have a lot of resources, so it just doesn't grow. So yeah, the sclerocactus people were not happy about that because these two rare cactuses are now one big, not rare cactus.
Ah,
Yeah.
gosh, the one that had a better benefit from environment would seem to be more rare. Was it? Is that? Yeah, that's
The
interesting.
one, yeah, the one that
The
the
little
big
guys.
angry one, it had just access to more water, more resources. It was on like often grew on hills, so maybe the aspect or the, the maybe it was less protected from wind than the ones that grew in the dry creek beds that were sandy soils and were just open to the elements. Maybe they just were just like, no, I'm just gonna chill. Like we're just really, really, really.
Feeling like a cactus.
Yeah, they're still trying to figure out this whole taxonomy thing using genetics instead of morphology, because morphology is not a great way to define species.
That is a really similar parallel in the fungal world where like things like rust, fungi, I think that cause disease on like wheat and barley. They also have a life stage that lives on, I think it's oak trees or some type of like, tree that grows next to the farm where they would, and until I think the early 2000s or the late 90s, mycologists thought that they were completely different, distinct fungi organisms from the way they look. But when DNA sequencing got, cheap enough to start to move into most labs and whatnot, they realized that this orange nasty thing that's growing on the crop and this gall that's growing on the tree are the same exact organism. And like, you know, they call it a telomorph and an anamorph. It's like two super, super different ways the fungal or the mushroom will present. Until recently, we thought they were different species. And it's like really just, is it ready to grow vegetatively or is it ready to grow reproductively? And this is how it presents, and it's still the same organism. Yeah, it's amazing how frustrating DNA sequencing has made the taxonomic science world, but it's really cool. Yeah, but it's
I know.
definitely
I like to
a
think
lot
of,
of updating.
what's that one giant plant that smells like rotting flesh?
The big
Morphus
flower,
titanium amorphous titanus or something like that. The one that
yeah, the
like
the
it's
cork
like this
flower, right, is
giant,
the common
yeah,
name.
yeah,
Yeah,
giant
yeah,
flower with a big penis in the middle. It like gives off heat when it blooms. It smells like rotting flesh. Have you seen the vegetative version of that?
yeah. I have
It
a cousin of that, Amorphophallus cognac. It's like the water tree, but yeah, it's like a little palm tree looking thing.
it's
It
like a
looks
palm
nothing like
tree.
like
Like,
yeah, yeah,
so
yeah.
I'm thinking about these people trudging through the, jungles of Borneo. Like, and they only bloom once every 7 to
Right.
15 years. So how often are you going to come across one of these plants in bloom? I would imagine not very often. Most of the time you're going to come across them in their vegetative state. And so you're going to see this palm tree and not realize that's the same thing. But yeah, like, so they may have seen that plant and not even realize those two things are the exact same thing. And then, to bring it back around to cannabis, cannabis sativa, cannabis indica, cannabis ruderalis, three species, but they're not. When we look at the
Right.
genetics, they're really just phenotypic plasticity plants that have been locally adapted to different regions, and really they still can interbreed. Yeah, same species, just different
Yeah,
morphology.
yeah. Like the host will be the host, but the environmental select and help it present and stuff. It's like that
Mhm.
old, you can take the boy out of the place, but you can't take the
Well, I think of like the track housing in like San Francisco. You've got all of these
Mm,
identical what look like they are identical floor plans, but you can change the facade, you can change the paint color, you can even change some of the interior a little bit where they all look different, but really they're all the same house. Like they're not any different at all.
sense. Yeah,
I
yeah.
kind of see plants as being similar to that.
I like it. Yeah.
All right,
Mm.
Mike, I think we're at the end of our time here today. Thank you so much
Okay,
for
right on.
joining me. Is there anything else you want to say, or tell everybody where they can reach you if they have questions about microbes, or the products that you have at Impello?
ImpelloBiosciences.com or on LinkedIn, anything like that. And yeah, I thank you for having me, Anna, Dr. Annabis. It's been a pleasure to, and after, 3 years ago, I think is the last time I saw you at
Yeah,
annual
yeah. Well,
conference.
maybe we'll come across each other at a conference sometime soon. My son still lives out there, so I'm home to catch up with him a few times, but yeah, no, not really.
All right. Well, I'm sure it's a lot of family time if you do come out and visit here, but Tess
Yeah.
is about 30, 45 minutes away from me, and I'm in Longmont. She's in like Thornton or Westminster or something. We should get coffee or something next time you're out
Yeah,
if
that'd
you.
be great. That'd be so awesome. All
Uh,
right, Mike, it was so good to talk to you. Thank you so much for your time. I appreciate you.
Likewise. Okay. It's your
Talk
Tuesday.
to you later. Bye.